Thank you very much for coming, and good morning. I'm Christian Pernstich, I'm the Research Director of Cell Guidance Systems, and I would like to introduce you to our sustained release growth for technology called PODS. And first things first, I would like to thank you, the people who did the work. That's our in-house PODS team, our collaborators from the Harvard Medical School, the originator of the technology, Professor Mori, and a hydrogel company in Holland, Bombyx. So growth factors, I'm sure you're all familiar with the basic problem of growth factors, their short half-life and the low bioavailability after a certain amount of time. And that's composed of consumption, but mostly due to the individual short half-life of the individual growth factor. Here I have two examples. One is standard soluble NIF. In the context of mouse ear cells taken from culture after a few days, and you see the rapid decline of the bioavailability. And that's measured with ELISA. And even further or steeper decline, GM-CSF in the context of TF1 cells, and again taken from culture and measured with ELISA, a fairly steep drop after 24 to 48 hours. And a number of solutions have been put forward in the past to overcome this problem, a sustained release. They range from mutations and chimera to stabilize each individual protein. They are more used in therapeutic protein delivery, but also embedding them as a depot formulation, if you will. And those biopolymers have been used like pegylation, PCL, PLGA encapsulation, and maybe more recently having them embedded in sheets and have layer-by-layer arrangements. And they all suffer from various problems, such as burst release, the bioactivity due to the embedding process can suffer. They're mostly not a platform because whatever works with one growth factor doesn't necessarily work with the other. The scalability is also sometimes a problem. Whatever works in the lab doesn't necessarily work at a larger scale. More problematic sometimes is the translatability in vivo. And I would like to introduce you to our protein nanodevice technology called PODS. I think we have found a very elegant solution to overcome many of those aforementioned problems. And by the end of this, I hope I could convince you that they are very much useful in vitro, 3D, organoid culture, stem cell differentiation, as well as easily translatable into in vivo as well for therapeutic protein delivery. Our protein nanodevices are made entirely of protein, and protein cages are not unusual in nature, and they range from chaperones and heat shock proteins to virus capsids of various sizes, shapes, geometries, all with the aim of protecting the cargo, whatever the cargo is, be it nucleic acid or be it other proteins. And people have, for the last 20, 30 years, tried to do that artificially by engineering those, having purified monomers, and then have them ideally self-assembling. And here's a very recent example of a fairly ingenious way of doing that by coordinating. You might see those little gold atoms here, have cysteines coordinated with gold atoms, and create self-assembling protein cages. Our protein cages are adapted from the life cycle of the Bombyx mori cypovirus, where in the late stage of the infection, there's plenty of variants already in the infected cell. Polyhedrin protein is produced on a large scale, and these polyhedrin proteins are self-assembling into regular lattices. Whilst the crystals are growing, protein crystals are growing, these virions are inserted via the turret proteins here on the surface. As you can see from an SEM image, those dimples or indentions, that's where the virions have been attached and in this case lost during the sample preparation. More importantly though, this X-ray crystallography image suggests it is not just on the surface, but it's all the way through the polyhedrin crystal. Professor Mori from the Kyoto Institute of Technology has found a way or thought that could be adapted to not only incorporate viruses, but any protein you like. He made a baculovirus construct where we have polyhedrin protein, which is necessary for the crystal growth, a cargo protein of your choice, and then an incorporation tag which attaches the expressed protein to the growing crystal. And we call it polyhedrin delivery system, PODS. And here you can see how they grow. It's all self-assembling in insect cells. And all we do is infect with baculovirus, let them grow, and then purify. The features and properties of those polyhedrin crystals, or protein nanodevices, are fairly remarkable. They withstand physical stresses like dehydration, freeze-thaw cycles, they're inert to temperature changes. They are inert against detergents and acids. The only condition or physical condition they dissolve is the alkaline. I'll come back to that in a moment. Their appearance is cubic, as you've seen and can see, and they're colorless, UV protective, and the last 2 points are more for in vivo. They are non-inflammatory and fully biodegradable. Again, it's just protein. In terms of handling in the lab, they're very robust. It's actually quite difficult to destroy them. They can be vortexed extensively. They don't dissolve in normal unsupplemented culture medium or in water. They adhere very easily to plastic, so you can coat tissue culture vessels, adhering them via centrifugation or simply pipetting on gravity settling and drying. Sub-aliporting is not needed for storage. So here we have experiments done with pods IL-6, stored in PBS over 6 months at various temperatures, 4 degrees ambient and 37 degrees, and then tested in a bioassay with TF1 cells, the bioactivity, and you can see there is not really much change over the time. And another example, again from LIF, in the context of mouse ES cells, and down here you can see without any LIF there is no proliferation of these stem cells. And if you store LIF, recombinant soluble LIF, at 4 degrees for 10 days, again, lost all its bioactivity. As a positive control, that's fresh recombinant LIF, and you can see that's the steep rise as you would expect it. And for our pods LIF, that's fresh, and that's stored for 5 months at 25 degrees for ambient temperature, and at 4 degrees, they're all fairly similar bioactive. Now, I've talked to you about incorporation, but release obviously is also very important, and what I said earlier, they're susceptible to alkaline conditions. That's because in the life cycle of the cypovirus, those crystals are being taken up by the next animal, passes through the stomach unharmed, and then in the gut, where it is pH 10, 11, that's where they slowly start to dissolve and release intact virions, which is not very useful in cell culture. That's why the other highlight is they are susceptible to proteases, and the sources of those proteases are cells themselves. They secrete MMPs, and depending on the cell line, more or less so. And serum has also an agent that is able to release the protease. Coming back to the very first image where I showed the decline of LIF and GM-CSF in culture, is exemplifying 2 things here. The first one is when you start your experiment with soluble, you put in your 100% and it declines with whichever rate individual protein has its half-life. With the pods, you have the exact opposite. Everything is sequestered inside the cubes. So you start off at 0, and you build up, depending on your culture system, the availability of your protein. Here, tracking over 5 days, and here over 6 days. Now, dissecting the GM-CSF a little bit further, again, with standard soluble, you have your rapid decline. And here we have plotted against, on the secondary axis, bioactivity of the protein available in the culture system. And you can see support of proliferation of TF1 cells is fine up to 3 to 4 days, and then bioactivity has been lost. The opposite is the case with our pods. You start off with 0, and you build up your reservoir of GM-CSF. And here plotting is the bioactivity and stimulation of proliferation. And as you can see, day 4 or 5, it really comes to its own. Since the released growth factor in this case is stimulating proliferation, which means more cells, which means more proteases, it is sort of self-regulating, almost a runaway train. Coming back to the sustained release, here we have in a cell-free, so there is no consumption, it is just decay of its own half-life. As you can see, again, we're starting off with 0, measuring ELISA, taking time points up to 7 days. Then a full media change, is resetting the system. So you remove all the released growth factor, you remove the accumulated proteases, and you start off with 0. Nevertheless, there is still in the crystals enough to come up again at day 14, full medium change resetting to 0, measuring at day 21 as a last time point. The data I showed you so far was in cultures where you have cells and pods, crystals next to each other, which is fine, but can create problems if you want to have a change of growth factor, something like differentiation protocols or reprogramming, where you would like to expose your cells, the stem cells, for a certain amount of time with one growth factor and then change to another growth factor. So you can do that, achieve easily by adhering those crystals to a tissue culture insert. And here we have the readout. If you have them next to the cells, that's the level of proliferation you can get with TF1 cells. And in the case of inserts, you are at, in this case, about 70% of that level. One reason being the distance between cells and so the source of proteases, and we rely on diffusion. So diffusion of the proteases from the cells to the crystals, and then of the growth factor from the crystals back to the cells. The protein nanodevices are also compatible with hydrogels, or hydrogel, and here I'm going to show you a very short video of pods embedded in a hydrogel and scanning through the Z layer from top to bottom. You can see a homogeneous distribution as they come in and out of focus. And the reason why that is, or could be advantageous, if you embed organoids, and that's from our collaborators, the Dutch hydrogel company Novacell, they've embedded breast cancer organoids in the hydrogel, and with the complete medium, that's the growth over 5 days they get of the organoids, comparing that to FGF2 pods. Here, if those are seeded in the well and then embedding the organoids into hydrogel and then spotting it onto the well, where you have the hydrogel layer sitting on the pods, fairly similar amount of expansion of those organoids. You can also embed pods into the hydrogel and have the organoids in there as well, so you have fairly short diffusion distance between the crystals and the organoids, and again, fairly similar outcome. So very compatible to that. The last example with organoids I'm going to show you from our collaborator at the Harvard Medical School. It's retinal ganglion cell organoids, and that's the differentiation protocol. And until day 20 is done with the specific media, and from day 20 to day 30, a single application of PODS BDNF and GDNF, which act as a chemoprotectant of those neurons, and only a half media change after 5 days, no further supplementation. And you can see when comparing it to no growth factor, we have about 40 to 50% increase of the total amount of RGCs produced. Going into a little bit further, looking at subtypes, much higher readout. Something that can only be achieved with normal soluble growth factor, but replenishing every 2 days. A further comment we had from them was that the organoids looked much healthier, looked smoother, and that has been attributed to less stress. So if you take them out, if you aspirate the medium and replenish with fresh medium, you subject organoids to a lot of physical stress. Whereas with pods, they are in the incubator for 5 days, you have a half media change, and another 5 days, so much less physical interaction. That, as well as with soluble growth factor, you have your massive spike at the very beginning and a trough after 24 hours or 48 hours, and then kind of massive spikes and troughs all the way through. So to sum up, PODS protein nanodevices are armored growth factor depots. It's a true platform technology, so we have produced at the moment up to about 90 different pod proteins, and we're constantly adding more. They are robust and a durable crystalline formulation. They're very easy to work with. I haven't actually talked about patterning or gradients, but as you can imagine, because they don't dissolve straight away, you can place them in one area of your culture dish. And if you then seed cells across, only the ones very, very close to the pods, crystals, see that growth factor. The release mechanism is adaptable. The more cells you have, i.e. the more proteases, the quicker they release, and the opposite is true as well. you can slow down release by either a secondary encapsulation or reducing the number of cells. The sustained release mechanism I demonstrated to you is ideal for organoid and stem cell culture because you have much less intervention, you have much less chemical spikes and chemical stresses. And a seamless transition possible from in vitro to in vivo.